quantum computers. So to start off, we're going to start with something that everyone has a little bit of experience with, and that's COVID. Big, bad, and scary. Nobody likes COVID. Boo! So world lockdown on March 2020. Then there was this whole vaccine development and certification thing, and then the vaccine was released in August of 2021. We don't care about the certification. That's boring. We're talking about the development. So like, what happened here? Like, we're going to use this as a little intro into RNA and like all that stuff. What happened here? So the COVID vaccine is an mRNA vaccine. Basically what that means is when you get your COVID vaccine shot, they inject a bit of RNA into you, and that tells your cells to make the little COVID spikes. Your cells make the little COVID spikes, and then your immune system learns how to fight that. And there are a few like key differences between DNA and RNA. This actually illustrates it perfectly. DNA is a double helix, whereas RNA is like a single helix. And RNA it's a messenger. It tells your cells what they need to do. So RNA is cool, but this is about RNA prediction. What is that, and what is RNA folding anyway? So essentially, because RNA is only a single strand, it's not a helix. To carry out its functions, it bonds with itself. So this is an RNA strand, and then it turns into this. It bonds with itself, and it pairs with itself. And this bonding and pairing creates a complex shape that we can identify as folds. And this is important because the RNA folds are strongly predictive of the function of the RNA. So if you know how the RNA folds, then you can tell what the RNA does. That was a quick refresher. So what is RNA base pairing prediction? So there are a few things that we have to just talk about really quickly. So RNA base pair prediction is an NP-complete problem. Basically what that means is it's really hard to find the solution, but it's really easy to check if it's right. An analogy I like is like if I gave you a pile of colored sand, and all the sand was different colors, and I told you to sort them. Very hard to sort, but it's easy to check. Like I could just look at it and be like, oh, that's sorted, no, that's not sorted. So the approach that people use is they essentially just randomize the sand until it comes out sorted, because it's very easy to check. So the current solving method is just running through all the possibilities and figure out which one is correct. This is done by using very powerful like computers, supercomputers generally, and fancy algorithms that try and make this a simpler problem. Because RNA prediction, the number of ways an RNA could, the number of possibilities for the bonds is equal to 2 to the power of the number of stems. And the stems, if we go back to that previous slide, here, these are each of the stems. So as you might imagine, that gets very quickly very complex. So these researchers had an idea. What if we use quantum computers? And now we talk about quantum computers. Yay! So what's the difference between a quantum computer and a normal computer? Well, a normal computer thinks in bits, which are a binary 1 or 0. Quantum computers are slightly different. They have things called qubits, where it's quantumly entangled particles that can represent a range from 1 to 0. Now the advantage of this is quantum computers have the potential to have far more processing power than normal computers, and be much more powerful. So this is a photo of a quantum computer. This study used a company called D-Wave, which basically rents out quantum computing time. And they have a quantum computer with up to 5,000 qubits, which is pretty high for quantum computers today. For reference, like a MacBook has 64 bits. So this study compares quantum computers from D-Wave to traditional methods of predicting RNA folds. And these are their results. Now this is a lot to unpack, so let's go over it like step by step. This is an RNA strand. As you can see, it loops back on itself, and this is the fold. It folds on top of itself. Each of these different colors are the base paired together. So purple is paired together, blue is paired together. This is the exact same thing, showing pairings. And these are 3 different methods of predicting the folds. EE is a type of classical computing method that is widely used. Same with REMC. It's another predictive method. And then the quantum computing. Quantum computing uses a process called quantum annealing, which is a type of algorithm for quantum computing for finding answers like this. And now this was graded on 2 metrics, sensitivity and specificity. And I always forget this. Oh yes, sensitivity basically points out correct answers. So how sensitive an algorithm is how good it is at pointing out correct answers. And specificity is how good it is at pointing out wrong answers. These were graded on 2 metrics, and as you can see, it actually got the exact same results. So this quantum computer was just as good as these 2 classical computing methods. And in fact, the quantum computer actually used less energy, which is really cool. But this is a relatively simple RNA strand. It has less than 45 stems. As far as RNA goes, it's not super complex. So they wanted to try it on something that was a little more complex. And for that, we have this data table, which is less than 45 stems and more than 45 stems. And as you can see, these are other types of predictive methods that are widely used today. As you can see, the quantum computer scored in the top range for both of these metrics. It's as good at like everything that classical computers can do. So like, what's the conclusion? What's like the impact of this? Basically, this study was able to show that quantum computing, even in its limited like baby stage today, is just as effective as classical computers. And of course, quantum computers are not widely available today, but as the technology progresses, as quantum computers become more powerful, this could have huge ramifications that we will talk about now. So current methods, because they can't deal with these giant amounts of data, like in like millions or trillions of possible solutions, they cut out important things that researchers may want to know, but that's like unavoidable. They just have a limited amount of processing power. They can't do it all. Quantum computers, on the other hand, could encode far larger amounts of data. So they could, an example that I've seen used multiple times is a quantum computer with 100,000 qubits, which has been predicted by multiple scientific studies as soon as 2030, would be able to encode the entirety of the COVID spike. And what this means is biochemists and researchers would be able to know exactly what the RNA does and how it does that. So going back to our COVID example, imagine like week 4 or 5, like knowing exactly how COVID works and what it does. Like these are the type of ramifications that we're talking about, which is just, it's incredible. Like this technology is still in its infancy. Quantum computers still only have, I think they top out like 6,000 qubits, but it's growing every single day. And as these become more and more powerful, like more and more things, there are more and more things that we could do with them. And this includes like encoding complex RNA structures, being able to know exactly what an RNA strand does and how it does it would be amazing. And yeah, this is what that study was able to show. It was able to show that this is not only feasible, it's like incredibly viable. It's an incredibly viable solution for a problem that like we can't handle massive amounts of data with our standard supercomputers. Yeah, thank you.